Rosalind Franklin was the first scientist to utilize X-ray diffraction to figure out the structure of DNA, which eventually helped Watson and Crick determine their own double helix model to be true. Her diffraction image was very important because it allowed for the double helix shape, represented by the X on photo 51, the radius, represented by the R, the pitch, represented by the P, and the number of phosphate molecules per turn, represented by the black dots, to be determined. While Watson and Crick ended up using her work without her permission, Rosalind Franklin is now universally recognized for her contribution to our present-day understanding of DNA.
So, along with Franklin's usage of X-ray diffraction that Aditya just talked about, the work of Chargaff allowed us to understand that DNA had an equal number of purines and pyrimidines, so that indicated that the two strands ran anti-parallel. And then Watson and Crick discovered that the AT bonds were the same length as the CG bonds. And so because of this, two key aspects of DNA replication were discovered. So one, that replication occurs via complementary base pairings. And then two, that replication was bidirectional since the two strands are anti-parallel. So DNA replication itself has several steps. So first, the DNA helicase unwinds and unzips the DNA by breaking the hydrogen bonds between the base pairs. And the DNA, or the gyrase, that reduces the torsional strain, which is created when the helicase separates and unwinds the DNA. Then to the template strand, the RNA primase adds an RNA primer. So that allows the DNA polymerase 3 to bind to the strands and then start that replication process. Then DNA polymerase 3 adds those nucleotides to the template strands in a 5-3 direction. So since the strands are anti-parallel and then the replication only happens in that direction, one of the strands, the leading strand, is going to be replicated continuously, and then the other one, so the lagging strand, is going to be synthesized in Okazaki fragments. So the lagging strand needs more primers since it's formed in fragments. Eventually, DNA polymerase 1 removes those RNA primers and then replaces it with DNA before ligase joins the Okazaki fragments together to form a continuous strand.
Some regions of DNA, called introns, do not code for proteins, unlike exons, but instead are utilized for other functions. One function is that these introns and their promoter regions are used in gene expression as they are the attachment points for RNA polymerase in mRNA transcription. This allows them to be involved in the process without actually being coded for. Additionally, in DNA profiling, non-coding regions are helpful because they are used in tandem repeats, illustrated here in each of these three people's profiles. And by using these tandem repeats, DNA profiles can be formed. Transcription is the process where the DNA is copied into mRNA by RNA polymerase before the process of translation. It occurs in a 5' to 3' direction. And it also uses proteins, enzymes, as well as complementary base pairing. When the mRNA is produced, the introns, or the non-coding regions, are spliced out by a spliceosome to leave solely the exons as the mature mRNA, which is then taken to be translated. Transcription can also be affected by environmental effects, especially in nucleosomes, which is studied through epigenetics. Overall, the main steps of transcription are initiation, elongation, and termination. Nucleosomes supercoil chromosomes by wrapping long strands of DNA around its octamer histone core, thereby protecting and packaging DNA. These histones also regulate the transcription of euchromatin during cell division. On one hand, by adding methyl groups, called methylation, transcription can be inhibited, while by adding acetyl groups to the DNA, called acylation, transcription can be promoted by binding the DNA looser to make it more accessible. While epigenetic tags can affect methylation acetylation, environmental factors can also do so by changing cell metabolism.
So translation has three main steps: initiation, elongation, and termination. So during initiation, the mRNA binds to the small subunit of the ribosome, which moves along the mRNA from the 5' to the 3' direction until reaching a start codon. A complementary tRNA molecule to that start codon will then bind to the P site of the ribosome, and a large subunit of the ribosome then binds to the tRNA in small subunit. So the next step is elongation. During elongation, a second tRNA molecule pairs with the next codon on the A site of the ribosome. And so now that the amino acid in the P site is covalently attached with a peptide bond to the amino acid in the A site, the tRNA in the P site transfers its amino acid over to the A site. The ribosome moves one codon along the mRNA strand in a 5' to 3' direction again, and the tRNA in the P site is moved to the E site and then released. And then the tRNA in the A site is now moved to the P site. Then another tRNA that's complementary to the next codon on the mRNA binds to the A site, and then this kind of transferring of amino acids repeats until you finally reach a stop codon. During the termination, which is this last step at the bottom, a stop codon has been reached and translation stops. And at this point, a release factor is attached to the A site. The polypeptide chain is released, and the ribosome complex disassembles, as you can see in this last picture.
So the last part of this unit was learning about the 4 levels of protein structure. So primary structure is the order and sequence of the amino acids which comprise the polypeptide chain, and it's formed by covalent peptide bonds that can be formed between adjacent amino acids. So that's pictured right here. Secondary structure is when the amino acids themselves fold or turn upon each other, and that's when they form two things, so either an alpha helix or a beta plated sheet, and you can see those right here. And this is because of hydrogen bonding between the amine and carboxyl groups of the amino acids that are non-adjacent. Tertiary structure is the way in which the polypeptide chain forms a 3D molecular shape, and that's because of the interactions between the R groups, and tertiary structure is super important for the function of the protein. So you can see that this is all one polypeptide, so like the background color is all gray, and then you have your alpha helices and your beta plated sheets. Quaternary structure describes a structure that occurs when multiple polypeptides or prosthetic groups form one larger protein. So that's where you can see there's like this pinkish color and the green color. So both of these could have the alpha helices and the beta plated sheets. A prosthetic group can also be a part of quaternary structure, and that's basically when an inorganic compound is involved in the protein. So an example would be the heme group in hemoglobin.